I have explained this slide earlier, so you can go over it again to know the components of ATP synthase and how the different parts function. The synthesis of ATP has been explained through what we call the binding change mechanism, which is based on observations suggesting that the substrate and the product binding properties of the active site could change as the protons moved across the membrane. The alpha 3 beta 3 oligomer of ATP synthase contains the three catalytic sites. At any given time, each site can be present in one of the three different conformations. Open, that's to say ATP can be released and ATP and inorganic phosphate can bind. Loose, bound ADP plus inorganic phosphate cannot be released. Tight, ATP is very tightly bound. So if you look here, we start with an open. As the protons enter through the proton channel, they will change the conformation of the open. It will become loose and it will become tight. That's the one shown in the yellow color, and that's true for the other. The loose side shown here in the brown color will become tight, then it will become open. The open, as you see, it releases ATP and it will be ready to bind ATP and inorganic phosphate. The formation and release of ATP can occur by the following steps, as I said, according to the binding change mechanism. ADP and inorganic phosphate bind to the open site. The rotation of the gamma shaft causes each of the three catalytic sites to change conformation. The open conformation becomes loose and the loose becomes tight. ATP is released from the open site and ADP and inorganic phosphate condense to form ATP in the tight site. The strongest evidence that ATP synthase is a rotating motor has been observed using the alpha 3 beta 3 gamma complex immobilized on a glass plate and modified by the attachment of a fluorescent actin element. You can find more details if you go through your textbook. As you have seen earlier, the synthesis of ATP takes place in the mitochondrial matrix. But this ATP, when synthesized in the matrix, it will leave the mitochondria to the cytosol where it will be used in different activities in the cell. So, when ATP is used in these activities, it will be cleaved to ADP and inorganic phosphate. So the ADP and inorganic phosphate must be transported back to the matrix so that ATP can be synthesized again. And there is exchanger in the inner mitochondrial membrane that will exchange ATP with ADP, as you will see in the next slide. Important to note here that ATP and ADP are transported in the free form. ATP has ATP4 minus, ADP has ATP3 minus. Recall earlier, I said that most ATP and ADP in the cell is in the chelated form. It's chelated with magnesium ions. But here, when it's translocated across the inner mitochondrial membrane, they are translocated in the free form. As you see here, there is a transporter in the membrane called adenine nucleotide translocase that will allow the entry of ATP 3 minus in exchange of ATP 4 minus, that is to say, this antiport transport. And there is also, we need an organic phosphate, which will also be transported through a phosphate transporter with H+. So this is a symport transport. What do we mean by the phosphate to oxygen ratio? This is the molecules of ATP phosphorylated to atoms of oxygen reduced. You have, as I said earlier, we need the 3 protons to be translocated for the synthesis of one ATP molecule. But we consume also 1 proton for the transport of the inorganic phosphate. So in other words, to synthesize one ATP molecule, we need 4 protons, 3 for the synthesis and 1 for the transport of inorganic phosphate. As you recall, I said when the electrons pass through complex 1, this will be accompanied by the transport of 4 protons across the membrane. In complex 3, again 4 protons. Sorry, there is a mistake here. 2 should be 4 and 4 should be 2 in the next complex. And complex 4 again, as I said, it will be accompanied by the transport of 2 protons. In other words, when NADH plus is oxidized to NAD plus, and these electrons are used to reduce oxygen to water, there are 10 protons bumped from the matrix side to the intermembrane space side of the membrane. As I said a while ago, that we need 4 protons to synthesize 1 molecule of ATP. So if we divide 10 by 4, this means that we end up having 2.5 molecules of ATP. So when NADH is oxidized to NAD plus, this will be accompanied by the production of 2.5 molecules of ATP. When QH2 is oxidized to Q, as complex 1 is not included, and as I said, complex 2 will not be involved in the production of energy. So we have only the 4 protons from complex 3 and the 2 protons from complex 4. So this makes the total of 6 protons. In other words, the amount of the energy formed when 1 molecule of succinate is oxidized to fumarate, or when QH2 is oxidized to Q, is equal to 6 over 4, and that is 1.5 molecules of ATP. Please note the correction in this figure. Under 3 should be 4 and under 4 should be 2. As you recall, when I discussed glycolysis, I said there is one oxidation reaction which resulted from the activity of the enzyme glyceraldehyde 3-phosphate dehydrogenase, whereby glyceraldehyde 3-phosphate was oxidized to 1,3-bisphosphoglycerate, as you see in this reaction, and I discussed that reaction in more detail when I discussed the glycolysis. This reaction was accompanied with the production of NADH. This NADH produced in the cytosol cannot be oxidized in the cytosol. As you know, it must be oxidized in the inner mitochondrial membrane. The problem, there is no carrier in the inner membrane for NADH. So how do we get the energy from this NADH formed in the cytosol? This is done through one of 2 shuttle systems. One is called the glycerol phosphate shuttle, and one is called malate aspartate shuttle, which I mentioned them earlier when I discussed the Krebs cycle, and I said I'll go over these shuttles in some detail later. The NADH formed in the cytosol will be used to reduce dihydroxyacetone to glycerol 3-phosphate, as you see in this reaction, catalyzed by the enzyme cytosolic glycerol 3-phosphate dehydrogenase. Now the glycerol 3-phosphate form will be oxidized to dihydroxyacetone phosphate by another glycerol 3-phosphate dehydrogenase complex, but this one is located in the inner membrane of the mitochondrion with its active site towards the intermembrane space. So the glycerol 3-phosphate will regenerate the dihydroxyacetone phosphate, but this complex has several electron carriers that eventually will be used to reduce Q to QH2. So the NADH formed in the cytosol now will enter the mitochondria as a QH2. In other words, when a QH2 is oxidized, we end up having 1.5 molecules of ATP, unlike NADH, which produces 2.5 molecules of ATP. So this shuttle will result in the loss of 1 molecule of ATP. And this shuttle is prominent in insect like muscles. This slide shows the reduction of dihydroxyacetone phosphate to glycerol 3-phosphate, catalyzed by the enzyme glycerol 3-phosphate dehydrogenase using the NADH, which was produced in the oxidative reaction of glycolysis, catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase, as we learned earlier. The second shuttle is what we call as the malate aspartate shuttle, and this occurs in liver and some mammalian tissues. What happens? The NADH here will be used to reduce oxaloacetate to malate, catalyzed by an enzyme cytosolic malate dehydrogenase. Now the malate will enter the inner mitochondrial membrane in exchange of alpha ketoglutarate through a translocator called dicarboxylate translocase. What happens in the matrix of the mitochondrion? Malate will be oxidized again to oxaloacetate, catalyzed by the enzyme mitochondrial malate dehydrogenase, and NADH will be released. Now, this NADH now will enter the electron transport chain and will be oxidized to produce ATP. So here there is no loss of energy. The NADH enters the mitochondria via malate, and the malate will be oxidized in the mitochondrion to oxaloacetate, releasing the NADH. Now, for this shuttle to continue, we need to export the oxaloacetate from the matrix to the cytosol again. There is no carrier in the inner mitochondrial membrane for oxaloacetate. So how is that done? This is done by a transamination reaction where oxaloacetate will be converted to amino acid aspartate through a transaminase enzyme. This is done a reaction between oxaloacetate and the glutamate to produce aspartate and alpha ketoglutarate. The aspartate formed now will be transported to the cytosol through a carrier glutamate aspartate translocase, and there in the cytosol another transaminase enzyme will convert aspartate to oxaloacetate. So that oxaloacetate now is regenerated and the shuttle will continue.